Hi everyone. So today our presentation is on the descriptive study titled Variation in Brown Rat Cranial Shape Shows Directional Selection Over 120 Years in New York City. So to start off, I'm going to give you guys some background on why this study is important. So the interest stems from how urbanization may change morphological traits within an environment. What morphological traits are outward appearance traits, and they could change from form and structure changes. So these changes are a result of selection pressures, and these selection pressures could be natural and artificial. So to give you guys some examples of these selection pressures, they could include increased temperatures, artificial lighting changes, infrastructure, increased noises, altered diets, and many other natural and artificial pressures. This is important because head morphology impacts how an animal interacts with their environment. So, to refer back on how urbanization may change morphological traits, the people in the study decided to look at Manhattan, New York, USA, because this is sort of a hub and the perfect example of urbanization. So this study really focused on investigating brown rat schools because these are very prominent in Manhattan. As you can see on the image in the right, they're showing many focused areas of rats in Manhattan, and there are just tons and tons of them. Below is an image of a brown rat school. So to give a little bit of background of brown rats, they were introduced to Manhattan around 1750, and since then their population has grown dramatically. We could assign this dramatic growth to how humans have actually grown in population as well, and with that there has been more of a food source, and so these brown rats have been able to increase in size, in body length, hind foot length, as well as distance from the supraorbital to nasal. So to start off and explain a little bit on how they conducted this study, the researchers used geometric morphometrics, and they compared cranial and mandible shape and size from rats that were collected from 1889 to 1895, as well as 2014 to 2016. So what did these researchers hypothesize? Their hypothesis was increased environmental complexity was the selection pressure for increased cranial capacity, which in turn increased fitness. They also had a null and alternative hypothesis. Their null hypothesis was genetic drift in gene coding for morphological variations can result in phenotypic changes. And then their alternative hypothesis is phenotypic plasticity explains selection for morphological changes in urban environments. So from this, they were able to come up with three predictions. Prediction A is that brain case size would increase over time for an increased cognition in a complex landscape. Prediction B is the nose would shorten over time to increase heat exchange given urban heat island effects. And prediction C is tooth row length would decrease over time as rats ate increased amounts of softer foods. And they basically observed these changes in cranial and mandible shape over time, and then they tried to test whether this was due to drift or selection.
First off, this experiment that was conducted in the article was a descriptive experiment, and this is so because they used careful observations and measurements to develop their findings. So the researchers just used info that already existed and actively manipulated a variable. So the first section is data collection, and here they talk about the samples of rats that they used in their experiment. The first source of rat samples was a collection of rats from the American Museum of Natural History in New York. And the second source of rat samples was a new collection of rats from the Yale Peabody Museum. Both collections only included adults with full eruption of third molars and rats with known sex. And this is important because the whole study is about the crania and the mandibles. So they use 3D geometric morphometrics to characterize the shape of the crania and mandibles. As you can see in the picture, that's kind of what the process looks like. And in this picture, and in the bottom one, you can see like the actual morphometrics for this experiment. So basically, the data collected was with a digitizer on what they call landmarks, and they took pictures of different angles of the crania and mandibles to create a single 3D configuration. And they used Morpheus for this. And missing landmarks were input as estimates by the software, and only the left side were accounted for, so they had to superimpose those images to create a symmetry of the entire crania and mandible to create these images. And on a side note, cranium and mandible sizes were calculated separately from the landmark configurations. The second section was error estimation. Through their experiment, 2 rats from each collection were measured about 10 times to estimate measurements of error, and they used ANOVA to calculate for these percentages. And the error values, as seen here, are dorsal with 1.8%, ventral with 1.9%, and mandible with 4.2%. So there were very minimal errors, which is a good sign for this experiment. Moving on to morphometric analysis, and here, so they tested for changes in the cranial or mandible shape in adult rats while controlling variation due to sex and size, and they did this with analysis of covariance and COVA, and they also use an F ratio test as their means of statistical significance. And this test was used because it tests the null hypothesis when 2 normal populations have the same variance. So the 2 populations being tested were quite similar. So that's why this type of test was handy. And they also quantified shape variation, and they also tested for change in size. Then we have our last little section, which is evolutionary mode. And here they were answering the question, what caused morphological shape change in both groups? So there were 2 possibilities, genetic drift or directional selection. It was tested by comparing phenotypic variance covariance matrices. So for context, in the article, they state that for mammalian skull evolution, the P matrix has repeatedly been shown to be a proxy for genotypic matrix, although it may be a poor model when trait heritability is low. So they ran this test and manipulated it then to get this bottom test, which was the D matrix, I believe, the divergent matrix. They ran 1,000 iterations for the multivariate drift test to simulate the amount of morphological divergence expected under the model of genetic drift, and they compared these values. And this is the equation that they used for that part.
In this table, we can see the results for the observed cranial and mandible shape with a size predictor. One of the more important factors on this chart consists of the R-squared portion, which measures variance. As we can see, the size of both the cranium and the mandible show the biggest R-squared, which demonstrates the highest variance. For the cranial shape, we can see that the significant changes were the centroid size, time period, and sex. As for the mandible shape, we can see that the 3-way interaction between log size, time period, and sex, and the 2-way interaction for the log size and time period demonstrated significant changes. In the second table, we are able to observe the size variance alone of the cranium and the mandible. Based on the values, we can see that there is no significant differences through time for either of the 2. For the evolutionary mode, we rejected the null hypothesis for shape change due to genetic drift for the cranium because the D matrix was greater than the confidence intervals of the simulated distribution, which suggested directional selection. We also failed to reject the null hypothesis for the mandible because the D matrix overlapped the simulated distribution. This suggested drift. In figure 1, we are looking at the changes in brown rat morphometric shape along with the PC axis. PC1 represents the allometric shape of the rat's head. Meanwhile, PC2 represents the time periods. The letters A through D show us the mean size shape of the heads. We are able to see a lateral and overhead example for each. The colors represent the different time periods. We can see that the black data is from the 1890s and the blue data is from the 2010s. The circle dots are female and the squares are male. Comparing the two time periods, we can see that there's a slight difference in the hindbrain case. The snout, upper molar teeth row, and a shift in the ear canal. Figure 2 compares the mandible shape, and the colors are consistent with figure 1. In this figure, we discovered that rats with larger crania have more curved and wider hind skulls. However, PC1 did not display any temporal variation. PC2 allowed us to see that the brain case did not increase over time. Another article had proposed that the brain case size may plastically increase as an initial response to invading urban environment, then decrease following acclimatization. However, our results can neither support nor refute this hypothesis because we only had data from 2 different time periods.
So for the first part, they looked at brain case and the nasal cavity sizes. So for the brain case, they found that it did not increase in the 120-year period. And they tried to make sense of that. They thought it would increase because of more complexity in the environment due to urbanization. But what they did is they looked at other studies. They found a study by Snellrod. And what he basically said was that because the brain for urbanization initially increases due to the changes, but then decreases due to acclimation. But this study can't really say anything to that because they only looked at 2 different data points. But what they did come up with is what they thought is that rats have to have less problem solving, and there's less complexity because they have to go less foraging to get their food. And that may explain why the brain case did not increase. They also talked about navel cavities being wider. Wider navel cavities are associated with colder temperatures. But so as you can see in the little picture, I showed a picture of a heat island. Their cities tend to be heat islands, so it's tended to be hotter. So a way to explain where it may be colder is they're thinking when they're breeding, the rats are like burrowing into the ground or they're going into sewers when they're having their babies. So I think that they did a good job trying to interpret their results, but the results, they couldn't make sense of it really using other studies. So I think that they could have suggested more research in these areas. Okay. Then if we're looking at the next 2, it's tooth row and mandible morphological change. So for the teeth, they found that the tooth row length was smaller. They found a decreased tooth row length, which is indicative of higher quality and or softer foods. I thought this part of the paper, they brought in a good amount of different research. And I thought it was really, really strong because it was research on other brown rats, which I thought was really important. And then also they found this higher quality diet. It could have been from being more carnivorous or more insectivorous. So I think further studies could be on that, but I thought that that was the strongest part in interpretation of their results. They then talked about mandible changes, which was possibly indicative of an increased diet. So basically they compared it to a paper, and the paper found 4 things. It was an increased carotenoid process, an increased angular processes, a lower incisor alveolar, and like increase teeth length. However, in this study, we only found 2 of those traits in these rats, which they did have an extended carotenoid process, and they did have a slightly increased incisor alveolar. However, I don't think that they could say that the hardness of the diet changed when they didn't find all 4 characteristics that the other paper found. And the other paper that they compared this to say the hardness in the diet was in mice and not in rats. And while they are relatively related, I just didn't think there was enough to really say the hardness in the diet increased. And I think it took away from the paper a little bit because they had such a strong case for the decreased tooth row length, and then they just didn't have the same evidence for the next one. So I want to talk about the limitations of the paper. They did say that some of their results or some of this directional selection could have been related to, is related to the influence of phenotypic plasticity. And we won't know how the environment has shaped these traits and to what degree necessarily. And then there was 2 other limitations. So urbanization, they looked at rats from the 1890s, but urbanization started in England well before that. So the rats were first in England in cities. Then they were brought here in New York City. And New York City started being really urban in like the early 1800s. So we aren't really looking at rats first being introduced to urbanization, which I think was a big limitation. And then also just there's been a lot of different changes in the 120-year period. Like there was World War I and II where food shortages. And then in the 1950s, there was a lot more food. And then also like the diets that we have has become more caloric, like increased in fats and sugars. And I think that probably could have influenced selection a lot. They did, though, at the end, suggest further studies and maybe looking at a city that's building up right now and seeing how rat morphology changes over time from a city that's just starting to maybe in 100 years. Okay, thank you guys. I hope you really enjoyed our presentation.